Leading a startup team, whether you're delivering a sugar rush, stocking coffee, or getting a regular delivery of snacks, Office Depot has solutions that fit every startup culture, from getting those first business cards and stationery to ordering fleece pullovers with your new logo. To learn how Office Depot and the California Technology Council have partnered to bring you savings on all of these startup essentials and more, go to californiatechnology.org/member benefits. I'm Daniel Levine, and this is the Bio Report.
Efforts to develop effective therapies to treat Alzheimer's disease have been stymied by a long history of clinical failures. Those disappointments have included two late-stage failures of AC Immune's Crenezumab, which the company is developing with its partner Roche. The two companies are continuing to pursue the drug under the belief that treating patients earlier in the progress of the disease will be critical. We spoke to Andrea Pfeifer, CEO of AC Immune, about the company's efforts to identify and treat Alzheimer's at its earliest stages, its efforts to develop a vaccine against the condition, and why she believes the future of Alzheimer's care will involve combination therapies similar to what's emerged in the area of cancer. Andrea, thanks for joining us.
Hello.
We're going to talk about Alzheimer's disease, AC Immune, and its efforts to improve outcomes for patients by identifying and treating patients early. Let's start with Alzheimer's disease itself. This is a neurodegenerative condition that is the leading cause of dementia and destroys a person's cognitive abilities. Why should we be concerned about Alzheimer's disease in a societal sense, and what's the potential impact of this condition on the population at large?
So I'm very happy to talk to you because I think the problem of Alzheimer is so broad and so important for society that basically on every level, which is the patients, the scientists, the government, media, should be concerned about it, because today we have 50 million Alzheimer patients. We have every 3 seconds, we have a new patient coming up. We are spending already 1 trillion on this disease, growing to 2 trillion in 2030, and there is no treatment. So I put on my banner the sort of the wish, the desire, the goal, the objective to leave a therapy and a diagnostic for this generation. Because if we are not dealing within this generation, then the impact, the cost, the pain is even bigger for the next generation.
Alzheimer's disease has been an area of great clinical disappointments. I think I saw a figure recently that suggested we've had 500 failed clinical trials since 2002. Is there something fundamentally wrong in the way the industry has thought about the targets it's going after to treat Alzheimer's disease?
I think it's a multi-component aspect in my view. I think you're right. There were many different clinical trials, but each clinical trial, actually, including our own, really contributed to the knowledge. I mean, we knew that earlier treatment is actually important because the later you treat, the more of these neurons are lost. And in fact, it is extremely important that you treat before neuron loss starts. And this is exactly where the field is going. What we have actually learned from these studies, that You have to diagnose early. You need a very early diagnosis in order to treat early, because only then you sort of catch the patient, the brain, before the loss is starting. So I think while it is quite difficult right now for the industry, for the scientists, for the scientific community, but in particular for the patients, I think all of this goes into a learning, telling us we need to have this early diagnosis, and we need to treat, potentially prevent a better blood building, and with that we might be actually successful. So everything what we call, and it's not politically meant, moves to the left, meaning to prevention rather than treatment. And honestly, if we can find a prevention, then that is the best possible outcome for everybody, because this means we keep all the neurons. This means that we keep the people living their lives and not being dependent on other people. So it would be the best possible outcome.
How is the condition diagnosed today, and how big a problem is that physicians are getting to patients too late in the progression of the disease?
Yeah, I mean, still, obviously, this is diagnosed by a neurologist, by psychiatrists, and usually they see the patient when the family or the patient himself or herself recognizes that something is not really in order, which means there are already mild cognitive impairment or even bigger cognitive impairment. So it's actually at a stage of a disease where a rather high irreversible loss of neurons has occurred, meaning you cannot reverse the disease. And this is really part of the problem. And this is, for me, if you like, if you ask me what is my biggest concern, what is my biggest objective, then it's really to find an early diagnosis, to allow an early treatment of the patients, and early means probably prevention.
Well, in that regard, what is AC Immune doing to identify patients at an earlier stage in the course of the disease so it can intervene when the best opportunity for having benefits from therapeutic interventions exist?
Yeah, so, well, the good news is that with the neuroimaging, a lot of progress has been made, and I think we are really looking into a situation right now where with neuroimaging, the risk factors, so a population, a patient which might have a high probability or a higher probability of getting the disease, can be identified. Also, for some of the neuroimaging, for example, the tau neuroimaging, there's a very nice correlation between the disease progression, the tau pathology, which you identify in these images, and finally the cognitive decline. So, in other words, we're starting to be able to look in the brain, and the biggest issue, of course, is in contrast to cancer, that you cannot just take a biopsy and look what molecular diagnostic you have. You basically need to look in the brain, and here, actually what we want to do is we want to look in the brain of people who still are healthy. So we call them preclinical, before they develop clinical symptoms. And obviously this completely excludes a biopsy. So what we are really trying to do, and AC Immune is really one of the leaders here, to develop imaging agents which allow you to identify these proteins which you see early. You see them 10, 20 years before, in fact, clinical symptoms come up. And some of them, as I just mentioned, the tau imaging really correlates extremely well with the clinical progression and might eventually become a surrogate marker, meaning we can actually test therapies in these patients and actually see how these biomarkers react, meaning we can come to biomarker-based clinical studies, which of course is much more exact than a clinical outcome, which is based on an assessment of a clinical or functional performance. So one is really neuroimaging. The other one is precision medicine, meaning what we learned over the last 2 years by some of the top scientists such as Virginia Lee and John Taranovsky from Penn University is that actually Alzheimer is not a mono-target disease. Alzheimer has plaques, Alzheimer has tau tangles, but Alzheimer has also other proteins. For example, TDP-43, which you find normally in ALS, or alpha-synuclein, which you find in Parkinson. So you find all of these different proteins in the Alzheimer brain, and obviously they influence the responsiveness, the response of the patient to the treatment. And I think one important aspect in this diagnosis is that, again, we have imaging agents against these different co-pathologies, which are sort of parallel to A beta and the second culprit, which is tau, and which potentially allows to really subclassify these people and actually treat them in a precision medicine way, as we have learned in HIV, in cancer, in other diseases. So my big hope is actually coming from the neuroimaging. My big hope is coming from other sensitive blood marker and CSF markers, which allow us to really 10, 20 years before the disease to identify the people at risk, and then treat the people according to their pathology. And I think if we do that, then we reach really this precision medicine approach, which has made us so successful in oncology. And that's really what we are doing. The last point to your question, and sorry for the long response, is of course that we are looking into genetic populations. For example, we are running a study in Colombia with Colombian population. They get Alzheimer very early, the 40 years, and 10 years later, they actually die of a disease. So while we have many symptoms and progression of the disease, which is very, very similar to a normal, a sporadic Alzheimer patient, the disease is much more defined, and the onset of the disease is much more defined. And actually, you can, and this is what we are doing, include the patients. Actually, they're not patients, the people, before they have the symptoms, which means we can test safe drugs before, in fact, these plaques are formed, or at a very early stage of the plaques, which means you can actually test the prevention of these plaques, and you can test potentially if Alzheimer can be prevented. In fact, the study in Colombia, which started in 2013, is probably it's really a landmark study, because we are looking at these people who get 100% sure Alzheimer, but we are looking at them before the disease starts, and we are deciding if, in fact, a safe drug, in this case, crenezumab, can actually prevent the onset. And if this is the case, this means Alzheimer can be prevented. And it comes back to my initial statement: the best possible outcome of all of these failures would, in fact, that we recognize the window when we can treat the A-beta monotherapy, and the window when we have to start to add combinations, for example, tau and others, and actually treat the people with a combination therapy, which is, again, not surprising, but surprising in Alzheimer, has been done for many other diseases, such as cardiovascular cancer every other disease. Why should it be different in Alzheimer's? So I'm actually very optimistic if you can really identify the windows for treatment in this genetic population and the other genetic population we are in fact investigating is Down syndrome, because here again in a very organized way, because the people have a triple chromosome 21, they all get Alzheimer's. They're 60 years, and of course it's a big concern to the parents of these children that they want to prevent this Alzheimer's disease in Down syndrome, because the children get older and get to an age where they get this disease. So for us, these populations have a multiple benefit. In one way, we are really helping this population to potentially not get the disease. We are learning from the treatment because we can start early, and hopefully we can identify biomarkers which will allow us to define prevention treatment for the larger sporadic population. So this work with this genetic population is extremely important for the larger population, and it's extremely important to decide when do we have to treat with mono or other combinations in order to actually prevent the disease rather than treating.
How much of a challenge does that become from a clinical trials point of view if you're dealing with a slow progressing disease that the onset may take 20 years for?
Yeah, this is exactly the point why, and it's a very good question, why we are dealing right now preferentially really with this genetic population, because as I mentioned to you, the disease progression is much faster in this genetic population, so everything happens much quicker, but what we believe right now in a very similar way. So by learning from the disease progression in this genetic population, we hope to identify actually biomarkers, risk factors, which would allow us to identify the people at risk. And there's a study by Keith Johnson who actually compared people of a certain age, 60 plus, years before they developed mild cognitive memory problems. And what he actually could show, and it's a very interesting study, that the people who had tau protein, same age group, they actually developed eventually years after, 5, 6, 7 years after, mild cognitive impairment, while the people, same age group, who did not have this tau protein actually did not develop the mild cognitive impairment. So in other words, we are at a point where there are certain biomarkers, and tau is certainly playing a very important role, which allow us to identify people at risk. And if this is the case, then of course our vaccine approach is the most relevant one, because it will be very difficult to treat people 10, 20 years before they are actually diseased, but it will be very possible to actually give one shot per year in order to prevent the disease. And this is why you actually see in AC Immune our broad portfolio, because we have antibodies in order to treat the tau and A-beta, alpha-synuclein, etc. But we also have the vaccination, because the vaccination will allow you to really preventively treat basically globally the population from a price perspective and also from an exposure perspective. And this is why we think the learnings will come from the antibodies, the treatment will come from the diseased population, from the antibodies, and recently now from small molecules, but preventively the vaccine will probably play the important role as long as we can establish the safety of these drugs, very importantly, and efficacy in this phase of the disease. So actually, Yes, it has been very difficult, but the learnings are extremely important to go now to the next step, which is precision medicine, to identify really the pathology of the people, subclassify the people, and treat them according to their pathology, and treat them early enough when the neuronal loss has not yet happened.
You have two platform technologies. I thought we could walk through them very quickly. The first is super antigen. What is it? How does it work? What does it do?
Yeah, the super antigen technology is actually a technology to generate antibodies and vaccine products. Now, I say it in a sort of simple way, but actually what these products do, they bind these proteins such as the beta plaques and the tau tangles in a conformation-specific way. Now, why is this so important? Both proteins and the other ones I mentioned earlier on are in fact self-proteins. And what we are trying to do is to overcome the normal immune system, which normally does not make antibodies against self-proteins. So we are inducing the immune system to make something which is normally not supposed to do. And not only that, we are making, in fact, antibodies which detect only the abnormal, the sick protein, but not the other one. And we are doing that by using actually liposomes. And on these liposomes, we on the surface, on the outside, we anchor via lipids the antigen, and we in fact have generated this technology that these antigens really mimic the pathological sick protein in the patient. In other words, if you give them this vaccination, either to mice or to people, the antibody response is very specific for the pathological, the sick protein. And with that, we reach three different benefits. Number one, we only generate these antibodies against the pathological protein, which means it's very safe and very specific. Number two, we don't waste, if you like, any antibody response. And number three, because it's done in a T-cell independent way, we don't induce T-cell toxicity, which was, and you might remember this, the problem with the Elan vaccine, which actually led to inflammation in the brain. We do not want inflammation in the brain. We have enough of that. And what we could actually show that our vaccine in our we are running multiple clinical studies, did not show any inflammation in the brain. In fact, the opposite. So that is really why we think we can give this vaccine to healthy people before we get the disease, because we have not seen any side effects which would make it critical for the healthy people. Now, on the small molecule side, the principle is the same, but it's, I would say, almost more difficult, because here it's not the immune system, so nature, which makes this molecule binding conformation structure specific. Here we have to basically design by really modeling these molecules, so these small molecules, which can actually once more bind only the misfolded protein and not the normal protein. And this again is important for efficacy, but also to avoid side effects. So, and this, in fact, this small molecule platform is got quite some attention recently. We made a very important collaboration with Lilly last year, because people were really claiming that it's impossible to make small molecules which can really bind so specifically the misfolded protein. And we could actually show that with a factor of 500 to 1,000 fold higher, we can really target this misfolded protein. And of course, the big advantage is that these small molecules, they get easily into the brain, they get easily into the cell, So actually we can inhibit the pathological process very much at the beginning of this process, meaning in the cell, in contrast to antibodies which work outside the cell. So we are actually quite excited about this small molecule platform as an addition to our antibody and vaccine work.
Do those drugs need to cross the blood-brain barrier, and is that any kind of an issue?
We do, and so for antibodies, we have actually shown that, for example, for our tau and A-beta antibodies, that an amount high enough to have an impact, at least in animals, can be achieved in the brain. There are multiple technologies right now out there which help to get more of these antibodies in the brain. And for the small molecules, this is really a nice feature. They are made for brain applications, so the majority of these compounds are made to go actually in the brain, and so we don't have to make special efforts and apply particular technologies to enhance this uptake in the brain. So here it's much easier.
You had talked about the trial in Colombia. Your drug Crenezumab had failed in 2 late-stage trials with Roche. What's the plan going forward? Is that the drug you're testing in Colombia now?
Yeah, I mean, Crenezumab, I mentioned to you before that the whole field is moving to the left, leaning towards prevention. And if you do that, then you need to more and more expose either healthy people or very early stage people with drugs, such as, in fact, these anti-A-beta antibodies. And it is extremely important when you treat healthy people that your drug is safe. And to date, Crenezumab is the only drug, at least to my knowledge, which did not show severe side effects, which would in fact prevent from treating healthy people or people with a very early stage disease. So one of the big benefits of Crenezumab is that it's a very safe drug, and it actually binds in specifically what we call oligos, which are supposedly the culprit of inducing the disease. Now, the right question is why, in fact, did Crenezumab not work in these pivotal trials, and why do we think it might work in the prevention in Colombia? So I think very importantly is we believe that in order to have a better monotherapy, so one molecule work, you have to treat very early, meaning pre-disease, pre-clinically. And in the Colombian study, we are actually treating these people before they get the disease. And as you know, they have a mutation, so they all will get the disease. And we think it's important what our studies say and what other people's studies say to actually go that early, because once the tau machinery is on, to just have an impact on the clinical benefit, on the clinical cognition, memory effects, the functionality by one therapy, for that the window might be very small. Now, having tau and other therapies available, once we know exactly when tau comes on, which by the way we are testing in this Colombian population, we are looking when tau comes on, which would allow us actually to add to the monotherapy tau and actually go for combination therapy. So in other words, this Colombian study is actually, for me, really key to the question, does a better monotherapy work if we treat the patient in this window before the disease starts, which we call preclinical, or in fact, do we need from the very beginning already to use combination therapy, for example, with tau? And both of these questions will be answered by the Colombian trial. So we are actually very, very excited, and everybody in the field is waiting for this study because it really will answer the question, is Alzheimer preventable, and then what would be the best way of doing that? And here, vaccination for me has a very important role to play.
Well, if you're successful in developing a prophylactic vaccine, do you expect this to be for the general population, or would this be for people with a specific risk profile for the disease?
For me, it's I would say it's almost like a question of health plans, and there are many political questions also coming into that. I personally believe that over time, and I mentioned already, tau potentially being such a marker. There are others in development which would in fact identify people at risk at a certain age. And I think if we are in a position to really identify this risk population, then I think it is almost a must, and every government in the world would need to think about it, how they can provide this vaccination to the people, because the concept of a subsequent cost linked to that, which is per patient around 200,000 or so today. We're speaking about 2 trillion costs to take care of these patients in 2030. I think it's so big that a preventive vaccination, which costs very little, should be made available. So yes, I'm looking at a global population, and I do believe that over the next years we will have the right markers available to identify these people at risk. And tau is certainly very high up on our list.
At the end of the day, you're still going after targets that other companies have failed with. What gives you confidence you'll be able to succeed where so many others have failed?
Well, I think so far the failures were around A beta. So, and again, the failures were around A beta tested in later stage population, and prodromal mild Alzheimer is a clinical population where you already have a big loss of the neurons. So we are moving from where all the failures were to a prevention situation, which is unique. And we believe that is already a very important move, and supported by the data. We actually, in contrast to now, I can really say that many of the players have actually diagnostic means. As I mentioned to you, we have imaging agents which allow us to look in the brain of the people for tau, for alpha-synuclein, for TDP-43 to really diagnose the patients in a precise way. So we are only treating the people essentially for the disease. And in fact, we are doing in particular trials around tau, and we will be the first company together with our partner Roche actually having a phase 2 study reading out in Q2 2020. So while A-beta sort of showed many failures, tau, it will be the first phase 2 study reading out in the field. So there is no failure yet in Alzheimer. And the other part, besides the genetic population, is of course combination therapy. I do believe that once we have this diagnostic means, diagnostic tools available, that selecting the patient and treating the patient with the right therapy is the way to go. And why should Alzheimer be different from cancer? I mean, I'm a cancer person. I worked on precision medicine in oncology. I think it's about time to apply the same principle which made oncology so successful.
Andrea Pfeifer, CEO of AC Immune. Andrea, thanks so much for your time today.
Thank you.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.